Hello, we are here again to talk on microbial anatomy and physiology. And today we have to discuss in detail the bacterial growth. We have studied the structure of microorganisms, organelles of microorganisms and their function, their composition. And next we have talked about the cell division, how cell bacterial cell division takes place. And today we have to talk bacterial growth. So this is growth curve. If you will see, we were talking here the microbial growth cycle. We have talked the batch culture, a closed system microbial culture of fixed volume, and a typical growth curve for population of cells grown in a closed system is characterized by four phases: lag phase, exponential phase, stationary phase, and death phase. So today we have to talk again the growth curve or growth phases. Here it is lag phase, where microbes are getting adaptation to the medium and taking some time. Whenever microbes are adapted to this environment, their growth is going to start rapidly. Rapidly, microorganisms are going to be divided and they increase their cell number, and that phase is called exponential phase. Here, growth is so fast because nutrients are fresh, waste products are less, and cells are at youngest and healthiest stage. And when here they are reaching an stationary phase, microbial growth is slow. The similar number of microbes are dividing and similar are dying, and it is almost at a stationary phase. Neither they are going to increase, neither going to decrease because nutrients are in limitation. Nutrients are limitation and due to excess amount and higher concentration of waste material, and nutritional concentration is going to reduce. And in death phase, here we can see it is going in decline because bacterial cells are going to die. Why they are going to reduce in number? Because concentration of waste material increased over here and the concentration of nutrients decreased over here. Therefore, bacterial cells went in stress and gradually they could not sustain to stress and they started to die. So this is called bacterial cell growth or growth phases. Lag phase, adaptation of microorganisms in nutritional media or in environment is called lag phase. Exponential phase or log phase. Bacterial cell number rapidly increases due to presence of fresh nutrients, higher concentration of nutrients, and the cell bacterial cells are in healthiest form, therefore they are growing rapidly over here. And here neither bacterial cells are increasing, neither decreasing, but they are at constant stage. And here bacterial cells went into stress because nutrients has been depleted, concentration of nutrients has been decreased, and concentration of waste material has been increased. Therefore, cells went into stress and they stopped further proliferation at this stage. So this phase is called stationary phase. After stationary phase, when nutrient concentration is reducing constantly and waste material concentration is increasing rapidly, so microbial cells are going to die. This is called death phase or decline phase. So this was the growth curve. And here we have to clearly explain that bacterial cell cycle is consistent on four phases: lag phase, log phase, stationary phase, and decline phase. And this is always studied in batch culture, which is a closed system. Because we are in closed system, we are not adding fresh nutrients, and neither we are harvesting. It is waste material, and it is considered as uh, calculated or measured in closed system. This is time scale, and here is bacterial cell number, log viable organisms per mm. Optical density we also can measure for this. So the microbial growth cycle we are discussing about it. Lag phase, lag phase interval between when a culture is inoculated and when growth begins. This is called lag phase. Interval between when a culture is inoculated and when growth begins. This is called lag phase. And exponential phase, cells in this phase are typically in healthiest state, therefore they are growing and they are dividing rapidly. And at the same time, nutrients are in plentiful and waste material is less, therefore bacterial cells are dividing rapidly. Stationary phase. In stationary phase, growth rate of population is zero. Either an essential nutrient is used up or waste product of organism accumulated in the medium. Two things can happen. Growth rate of population is zero. Why it is zero? Because either an essential nutrient is used up or waste product of organisms accumulated in the medium. Either concentration of waste material produced by microorganisms has been increased or nutrient concentration has been reduced. Therefore, microbial growth rate is zero. Neither they are going to divide, neither they are going to die, and they are in stressful condition. Growth medium. So yes, the microbial growth cycle death phase. If incubation continues after cell reach stationary phase, the cells will eventually die because nutrients has been limited or has been finished. Waste concentration of waste material has been increased, and these two conditions exerting uh, stress too much on microbial cells, and eventually they are starting to die. Temperature and microbial growth. Now we have to see what is the role of temperature on microbial growth. So we will see effect of temperature on growth, microbial, as well as we will see microbial life in cold environment, as well as microbial life in high temperature. So here we can see that This is the temperature and this is growth. When microbial growth is there, gradually when temperature is increasing, microbial growth is also increase. There is a time at this stage when temperature is optimum, when temperature is too high, microbial growth stops and it is going to reduce directly. So what happens? When the when temperature is low, enzymes reaction occurs in increasing rapid rate. When temperature is normal, it is near to 37 or optimal temperature. Enzymatic reactions occurring in increasingly rapid rate. Okay, what happens when temperature is too high? Enzymatic reactions occurring at maximal possible rate. It is optimal because microbial enzymes are working at optimal rate over here. When temperature will increase more than optimum, it is activity will be stopped. So what will happen at this stage? Potent denaturation, collapse of the cytoplasmic membrane, and thermal lysis. When temperature will increase from this optimum temperature, what will happen? Microbes will not tolerate at this temperature. Why they will not tolerate? Because proteins denaturation will start. Collapse of cytoplasmic membrane will take place and bacterial cytoplasm will lyse and they will not survive. So what happens when temperature is low? Membrane gelling transport process so low that growth cannot occur. When temperature is too low, microbial growth is also slow because microbial enzymes are not working optimally. So this is growth. This is the mechanism, kinetics of the growth curve. All enzyme reactions occurring at increasing rapid rate at this temperature. So effect of temperature on growth, microorganisms can be classified into groups by their growth temperature optima. Number one, sacrophilic microorganisms, sacrophiles, class number one. Microorganisms that can grow at very low temperature, those are called sacrophiles. Mesophiles, microorganisms who can grow, who can function at mid-range temperature are called mesophiles. And the thermophiles are those microorganisms which can which can function at high temperature. Those are called thermophiles. Hyperthermophiles are group of those microorganisms which can work at extreme high temperatures are called hyperthermophiles. So on the basis of temperature range, microbial groups has been divided into four groups. Sacrophiles microbes can grow at low temperature. Mesophiles microbes can grow at mid-range temperature. Thermophile microbes can grow at high temperature. Hyperthermophile microbes very high temperature. So we can see it in the cell. This is growth rate and this is temperature, maybe -4 to 10. These are called sacrophiles. And example of microorganism is Pleuromonas vacuolata. These are microorganisms can grow at 4 degrees C. And these are sacrophiles. So microorganisms can grow at this range from -to 10 degrees C -5 to 10 degrees C, and maximum temperature is for the growth takes place are sacrophiles. And about mesophiles, the example of mesophilic is Escherichia coli, and this ability to grow from 10 degree to 45 degrees C, and optimum temperature for this microorganism is 39. So this is called mesophilic microorganism. Thermophile microorganism example is Geobacillus stereothermophilus. The range of temperature for this microorganism is 40 degree to almost 70 degrees C, and maximum temperature and most optimal temperature is 60 degrees C, where its growth takes place at high rate. And hyperthermophile microorganisms example is Thermococcus ciller. Temperature ranges from 65 to 95 degrees C, and optimum temperature for this type of microorganism is 88 degrees C. And hyperthermophiles example is Pyrolobus fumeri, and range of temperature for this microorganism is 90 to almost 115 degrees C. So these are the different classes of the different groups of the microorganisms on the basis of the temperature. So we can see these microbes has been divided into sacrophiles, mesophiles, thermophiles, hyperthermophiles, and hyperthermophiles. So from this slide and from this picture, we can conclude, we can say that microorganisms are present at every corner of this globe, either where temperature is very low, either temperature is very high. Maybe number of microorganisms can be less or high, and at moderate temperature where temperature range is 39, where most of the plants and animals can grow, here microbial population can be more, and at extreme regions it can be a little bit less. So effect of temperature on growth, mesophile organisms that have mid-range temperature optimum found in, they are living in warm-blooded animals in alimentary canal and outer skin of the warm-blooded animals. Mesophiles are also normal flora of warm-blooded animals as well as terrestrial and aquatic environments, temperature and tropical latitudes. So mesophiles can live over here at this range. Mesophiles are found in uh, normal flora of warm-blooded animals, found in aquatic environment as well as in terrestrial environment, temperate and tropical latitudes. So number of mesophiles or microorganisms is too high. They can be found in most ecological needs of this globe, because in most ecological needs of this globe have mid-range temperature. So microbial life in cold, how microbes are living and which type of microbes are living in cold environment, if you will see. In cold environment, mostly microbes are living are called extremophiles, organisms that grow under very hot or very cold conditions are called extremophiles. Extremophiles, organisms that grow under very hot or very cold conditions are called extremophiles. And sacrophiles, organisms with cold temperature optima, inhabit permanently cold environment. Inhabit permanently cold environment. Okay, saprophytes are called those microorganisms which can live at very cold temperature, inhabit permanently cold environment. And sacro-tolerant organisms that can grow at 0 degrees C but have optima of 20 degrees C to 40 degrees C. More widely distributed in nature than saprophytes, these are called sacro-tolerant. Sacro-tolerant are those organisms that can grow at 0 degrees C but have optima of 20 degrees C to 40 degrees C, more widely distributed in nature than saprophytes. Microbial life includes microbial molecular adaptation to saprophytes, production of enzymes that function optimally in the cold, features that may provide more flexibility. So it is a question that how microbes can survive at very cold temperature. So we should understand that how microbes can survive at very low temperature. So microbes which are living at very low temperature, they are producing certain enzymes that function optimally in the cold. Features that may provide more flexibility. So we are going to stop over here, and in next session we will talk from here. You have to go through book for understanding of this chapter. Thank you. Have a nice time, please.